Ernest Duchesne. You've never heard it. Almost nobody has. And that is exactly the point. In 1928, a Scottish bacteriologist named Alexander Fleming looked at a contaminated petri dish in his London laboratory and saw something that would change the history of medicine forever. He saw mold-killing bacteria. He called it penicillin. The world called him a genius. He won the Nobel Prize. Statues were built. Textbooks were written. His name became synonymous with one of the greatest medical breakthroughs in human history. That story is true. But it is not the whole story. Because 31 years before Fleming's petri dish, in a military laboratory in Lyon, France, a 23-year-old doctor had already seen the same thing. He had run the experiments. He had written the proof. He had submitted it to the most prestigious scientific institution in France. And they never wrote back. His name was Ernest Duchesne. He discovered the antibacterial properties of penicillin mold in 1897. He died in 1912, 16 years before Fleming's discovery. Unknown, unrecognized, and forgotten. He was 36 years old. To understand what Ernest Duchesne discovered, you first need to understand what the world looked like before it. Not the world of warfare. Not the world of politics. The world inside a wound. In the second half of the 19th century, surgery had advanced considerably. Anesthesia existed. Surgeons could now perform procedures that would have been unthinkable decades earlier. But there was a problem. Patients survived the operation. And then they died anyway. Infection killed at staggering rates. Following major surgical procedures in the 1860s and 1870s, hospital mortality rates from post-operative infection ranged from 30 to 60% in some institutions, according to contemporary medical records. A soldier shot in the arm didn't die from the bullet. He died from what grew inside the wound afterward. Gangrene, septicemia, erysipelas. Words that meant, in practical terms, a slow and agonizing death, often within days. Germ theory, the understanding that microscopic organisms caused disease, was still finding its footing. Louis Pasteur had published his foundational work in the 1860s. Joseph Lister had introduced antiseptic surgical technique around the same time. Progress was being made. But it was grinding, slow progress, measured in decades. And while scientists argued over methodology and institutions debated theory, people continued to die of infections that a simple compound might have prevented. It was into this world that Ernest Duchesne arrived. Born in Paris in 1874, Duchesne enrolled at the École du Service de Santé Militaire, the military medical school in Lyon, in the early 1890s. He was, by all historical accounts, an exceptionally methodical student. Precise, curious, persistent. And he was paying very close attention to something that everyone else was walking past without a second glance. The Arab stable hands at the military barracks had a habit. When horses developed sores from the friction of saddles, raw, open wounds vulnerable to infection, the stable hands would pack those wounds with mold scraped from the damp saddle leather. The wounds healed. The stable hands couldn't explain why. They didn't need to. It worked. It had always worked. It was simply what you did. Duchesne watched. And he began to ask a question that nobody around him thought to ask. Why? Ernest Duchesne was 22 years old when he began the experiments that would define his life, and ultimately consume it. His resources were modest. His laboratory space was limited. His time was constrained by the demands of military medical training. None of that stopped him. The substance Duchesne focused on was Penicillium glaucum, a common mold, the kind that grows on old bread, on damp leather, on forgotten fruit. It was everywhere. It was considered, essentially, a nuisance. Duchesne believed it was something else entirely. His experimental approach was systematic and, for its time, remarkably rigorous. He introduced cultures of Escherichia coli and Bacillus typhosus, dangerous bacteria, into controlled environments alongside Penicillium glaucum mold. He observed what happened. The mold inhibited bacterial growth. In some cases, It stopped it entirely. He then tested the compound on living animals, guinea pigs, injected with lethal doses of typhoid bacilli. The animals treated with penicillium cultures survived. The untreated control animals died. He documented everything. His 1897 thesis, Contribution à l'étude de la concurrence vitale chez les microorganismes, Antagonism entre les moisissures et les microbes, translated roughly as Contribution to the Study of Vital Competition in Microorganisms, Antagonism between Molds and Microbes, laid out his findings with clinical precision. The language was careful. The methodology was sound. The conclusions were clear. Mold could kill bacteria. Specifically, and this is the detail that stops historians cold, the same mold that would later make Alexander Fleming famous. Duchesne was 23 years old. He had, in a modest military laboratory in Lyon, with limited equipment and no institutional backing, produced experimental evidence for one of the most important medical discoveries of the century. He packaged his thesis. He addressed it to the Institut Pasteur in Paris, the foremost center of microbiological research in the world, founded by the man whose name defined the science. And he waited. The Institut Pasteur did not respond. Not a letter. Not an acknowledgement. Not a rejection. Silence. Historians who have examined this episode offer several explanations, and none of them are entirely satisfying. Some suggest that Duchesne, as an unknown military student with no scientific reputation, was simply not taken seriously. The institute received dozens of submissions. Priority was given to established researchers. Others point to the age factor, a 23-year-old claiming to have found a microbial antagonist capable of killing lethal bacteria would have seemed, to the scientific establishment of the 1890s, improbable at best. Some historians believe the thesis was simply never read. The exact reason remains unknown. What is known is this: Ernest Duchesne graduated, continued his military medical career, and never returned to his penicillin research. Not by choice. Shortly after completing his thesis, Duchesne was diagnosed with tuberculosis, the disease that would eventually kill him. His health declined steadily. The research that might have defined a century was set aside, first out of necessity, then out of time. He died on April 12, 1912. He was 36 years old. His thesis sat in an archive. The Institut Pasteur, the scientific establishment, the medical world, none of them had ever acknowledged his work. 16 years later, in a London laboratory, a Scottish bacteriologist named Alexander Fleming would look at a contaminated petri dish and see exactly what Duchesne had seen. The world would celebrate Fleming. Duchesne's name would not appear in the celebration. It would not appear anywhere. Alexander Fleming returned from a summer holiday on September 3, 1928, to find that one of his bacterial cultures had been contaminated. A common enough occurrence in any laboratory. What was uncommon was what he noticed next. Around the mold contamination, a halo. A clear zone where the bacteria had died. Fleming examined it. He identified the mold as belonging to the Penicillium genus. He wrote in his notes, "The mold was therefore acting as a germicide." He published his findings in the British Journal of Experimental Pathology in 1929. The observation was, in its essential form, identical to what Duchesne had documented 31 years earlier. The same mold. The same bacterial inhibition. The same conclusion. Was Fleming aware of Duchesne's work? Historical records do not provide a clear answer. Fleming's published paper does not reference Duchesne. There is no evidence in surviving correspondence that Fleming had read the 1897 thesis. Given that the thesis was written in French, published in a limited military medical context, and never formally recognized by the wider scientific community, it is entirely plausible that Fleming had no knowledge of it. Entirely plausible. But the question, once asked, is difficult to set aside. What happened after Fleming's discovery is its own complicated story. Fleming himself did not immediately pursue the clinical application of penicillin. It would take another decade, and the work of Howard Florey and Ernst Boris Chain at Oxford, to develop penicillin into a usable antibiotic. Fleming, Florey, and Chain shared the Nobel Prize in Physiology or Medicine in 1945. Ernest Duchesne received no mention. It was not until the late 20th century that French historians, reviewing military medical archives, rediscovered Duchesne's 1897 thesis and began to argue, carefully, methodically, that the record of penicillin's discovery was incomplete. In 1949, the Institut Pasteur quietly acknowledged that Duchesne's work had anticipated Fleming's findings. It was not front-page news. It was a footnote. There are those in the history of science community who argue that independent discovery, two researchers arriving at the same conclusion separately, is common enough that Duchesne's case, while tragic, does not diminish Fleming's achievement. That argument has merit. But it does not answer the harder question. Not, who discovered it first? But, what happened to the people who died between 1897 and 1928, while the proof sat unanswered in a Paris archive? Between 1914 and 1918, the First World War killed an estimated 17 million people. Of those deaths, a significant proportion were not caused by bullets or shells. They were caused by infection. Wounds that became infected. Amputations that turned septic. Simple lacerations that, in the mud and filth of the Western Front, became death sentences. Military historians and medical researchers have estimated, conservatively, that effective antibiotic treatment could have saved hundreds of thousands of lives in that conflict alone. Penicillin, had it been developed from Duchesne's 1897 findings, might have been available in clinical form by the early 1900s. It was not. The gap between Duchesne's discovery and Fleming's, 31 years, is not merely a historical curiosity. It is a measurement. A measurement of institutional failure. Of the cost of silence. Of what happens when a scientific establishment decides, without reading the evidence, that a young unknown researcher has nothing worth hearing. This is not an isolated story. The history of science is filled with researchers whose work was ignored, rejected, or absorbed without credit, often because they were the wrong age, the wrong nationality, working in the wrong institution, or simply existed before the world was ready to listen. Ignaz Semmelweis, the Hungarian physician who proved that hand-washing prevented maternal deaths in childbirth, was ridiculed by the medical establishment, committed to an asylum, and died before his ideas were accepted. Gregor Mendel's foundational work on genetics sat largely unrecognized for decades after his death. These are not accidents. They are patterns. The sociology of scientific recognition is a field that researchers have studied with increasing rigor. What the evidence suggests, consistently, is that scientific institutions, like all human institutions, are subject to the same biases that govern human judgment everywhere. Prestige matters. Age matters. Affiliation matters. Nationality matters. The quality of the work, on its own, is rarely sufficient. Duchesne had the quality. He did not have the prestige. And so the drawer closed. And the years passed. And the soldiers died. Ernest Duchesne is buried in Paris. His grave does not attract tourists. There are no statues. No streets named after him. No hospital wings bearing his name. In France, a small number of historians and medical archivists know his story. Outside of France, almost no one does. In the years after his rediscovery, Duchesne has received modest, belated recognition. The Institut Pasteur, the institution that never answered his letter, formally acknowledged his contribution in retrospective historical assessments. Some French medical historians refer to him as the true discoverer of penicillin. Others are more careful, noting that discovery without development is an incomplete equation, and that Fleming, Florey, and Chain transformed an observation into a medicine that saved hundreds of millions of lives. Both positions have merit. Neither brings Duchesne back. The question that Duchesne's story leaves open is not really about him. It is about all the others. History does not have a reliable mechanism for recovering what it loses. Archives burn. Institutions move on. Names are forgotten because forgetting is easier than remembering, and because the story of the winner is always simpler than the story of the person who came first. How many other Ernest Duchesnes are in those drawers? How many other theses sit in military archives, in university basements, in the collections of institutions that never wrote back? Alexander Fleming deserves his place in history. The development of penicillin was one of the most consequential achievements in the history of medicine. By the late 20th century, derivatives of Fleming's original discovery had saved an estimated 200 million lives, a number that continues to grow. That is real. That is extraordinary. That belongs to the record. But the record is not complete if it begins in 1928. Local accounts from the military academy in Lyon describe Duchesne as a quiet, serious young man, the kind of student who stayed late in the laboratory and asked questions that made his professors uncomfortable. According to surviving documents from his academic record, his thesis was considered thorough and technically accomplished. It simply was not considered important. In the end, what history chose to remember about penicillin is a story about a lucky accident, a contaminated petri dish, a curious scientist, a discovery that changed the world. That story is true. But underneath it, in a French military archive, in a thesis submitted by a 23-year-old who had already seen what Fleming would one day see, there is another story. One about a man who was right too early. About an institution that didn't listen. About the distance between a discovery and a recognition, measured not in miles, but in decades. And about all the lives that lived and died in that distance.